RSSL have been on campus in Reading for over 30 years now, which puts us really close to the golden triangle for cell and gene, which is, Oxford, Cambridge and London, where all the research is coming out and being developed. As we've expanded over time, we've added labs organically and added to the teams, just in response to our customers' needs. In terms of our offering for cell and gene, this starts with everything from raw materials going into production. That could be simple buffer salts, complex mixture. Specifically though, we've expanded into the biological raw materials, so this is all the growth factors and cytokines required to keep the cells happy. After that, we would look at the biosafety, so are the products sterile, because they have to be sterile to go into the manufacturing for cell and gene. Obviously they have to be sterile except for the cell components to go back into the patient. So we offer various levels of biosafety. It could be bioburden, it could be endotoxin, and it could be sterility testing based on the needs of the customer. When it comes to product-specific characterization, that really, again, is based on our customer's needs. So it could be simple IDs, and this could be straightforward. It could be something like mass spec, could be an ELISA if it's a biological, but it could be a chemical test as well, so that's needed. For stability, we offer as our customer needs. So we could be performing in-use studies, which is how long once they're in the bags, and ready to go for the cell events, for the transformations for cell and gene therapy, are all the constituents stable for, or we could go for how long are they stable long term. Alongside the laboratory testing, we also offer quality training, so everything around the quality needs, whether that's QP training, whether that's GMP. We have specific modules for ATMPs and cell and gene as well, so really we're looking after the product, around the product, and then the quality systems of our partners, so every aspect of quality around the site as well. All of our services are at one site or several interconnected buildings on the same site. So this means that when it comes to troubleshooting, you've not just got a team of 5 who would say, this is an ELISA problem, let's view the issue as an ELISA issue. It could be a mass spec issue, it could be an impurity, and we can bring that expertise all together very rapidly without having to go to cross-site, cross-country, cross-nation collaboration.
Responsive.
Customer-focused.
Innovative.
Friendly.
Knowledgeable. Flexible.
So when you bring all these various aspects of RSSL together, it really is all about science with service.